Hi, my name is Bimo, and in this video I'm going to show you what research I've done for the past 10 years. My research has been revolving around peptide for therapeutic purposes. I have done some research on peptides for industrial purposes, but due to time limitation, I will not cover the topic in this video. So, since this is a summary of 10 years research, please bear with me and thank you for sparing your time for listening to this presentation. Peptide is the polymer of amino acids. There are 20 amino acids that can be arranged into almost unlimited number of peptides in different lengths and properties. This is what makes peptide interesting. You have flexibility in your hands to create millions of molecules with different biological properties just by combining 20 building blocks. Creating peptides is just like creating a building using Lego blocks. The only limit is your imagination. Another reason why I choose peptide as my research, and after this presentation I do hope I successfully ignite your interest into peptide research, is that peptide synthesis is easy. 50 years ago, Bruce Merrifield discovered an amazing way to synthesize peptides using solid phase system. This method tremendously cuts the time for synthesis and purification steps needed to obtain pure peptides. When I was in UPM, my students can obtain the pure peptide in just 2 weeks. If you buy the peptide from any company, you can get your peptide in 3 weeks, including shipping process. So I can proudly say that I was able to train my students, so my lab is in par with any other labs in terms of productivity. Another amazing thing about peptide is that you can get significant biological activity just by using a 3 amino acid peptide. Like in this slide, you can see an RGD peptide, which has been used in removing blood clot inside human blood vessel. So this peptide has only 3 amino acids. If you want to go the extra mile, you can synthesize longer peptides. For example, in this slide, you can see fusion. It's a 36 amino acid peptide that has been used as an anti-HIV drug. Or if you want to really challenge yourself, you really want to challenge yourself, you can synthesize up to 100 or 150 amino acid peptide, and many interesting things can be observed because basically you are synthesizing a protein. If you are interested in doing business, peptide is something very promising to be looked at. 3 years from now, the global peptide drug market is expected to increase to 25.4 billion US dollar. And currently there are 60 US FDA approved peptide medicines in the market, with close to 150 peptides in clinical trials, and more than 500 peptides are in preclinical development. If you look at this figure, small molecules are still dominating the distribution of the drugs approved by the FDA. But you can see that peptide has taken 8% of the drug market share, where natural products surprisingly only take 6% of the drug market. If you see this progress from perspective view, where 20 years ago, people will laugh at you when you propose peptide as drug, you can see that the future of peptide drug is very bright. Well, I've shown you plenty of good things about peptide drug. I think you might want to jump into this research field right now, but please not too fast. As I have done research in this area for about 10 years, I can tell you that there are a lot of challenges in producing therapeutic peptides. First, you cannot design a peptide out of the blue. You need to really carefully choose the correct sequence to ensure that your peptide is biologically active. You need some preliminary experimental data here. Second, you need to measure how active your peptide is. There are various methods that you can choose from. Mostly are biophysical methods, such as NMR, fluorescence, SPR, ITC, and molecular modeling. In this video, I will only focus on NMR and molecular modeling. The first part of my talk will cover the work that I did during my postdoc in the University of Kansas. I was tasked to synthesize and study the peptides that inhibit the interaction between ICAM1 and LFA1 receptors. This is a little bit introduction about our immune system. When our body is challenged by invaders, our immune system gets activated by two signals, called signal 1 and signal 2. These two signals are used by antigen-presenting cell or APC and T cell to communicate before generating host immune response. Our hypothesis was if we can inhibit signal 2, then the T cell adhesion will be modulated and result in the inhibition of human response. This approach is very useful in designing inhibitors to suppress autoimmune diseases and allograft rejection. In my work, I use a cyclic peptide called CIBR. This 12 amino acid peptide was discovered in mid-90s by Professor Xian's group and has been used ever since in autoimmune disease research. The sequence of CIBR peptide was taken from domain 1 region of ICAM1 molecule on the surface of APC. This peptide is thought to interact with LFA1 molecule on the surface of T cell. The structure of CIBR is known to adopt beta turn structure at PRGG sequence, proline, arginine, glycine, glycine sequence. The structure was known in 1999, but how this peptide binds to LFA1 was unknown at that time. When I came to Xian's group in 2005, we worked to study how this peptide interacts with LFA1 molecule. By using monoclonal antibody cross-inhibition methods, we discovered that this peptide binds to two regions in LFA1 receptor, which are L site, blue in this slide, and Midas, red in this slide at the top of the molecule. However, we thought it would be a good idea to confirm our finding by using different techniques. So, through collaboration with Blanco's group in Spain, we use high-resolution NMR to study where CIBR peptide binds to LFA1 receptor. We use C13 and N15 label LFA1, and then we discovered that CIBR peptide binds to L site. And this result confirms what we discovered previously using monoclonal antibody cross-inhibition method, even though we did not see the peptide binds to Midas site, which is at the top of the LFA1 receptor. We didn't see that. All right, then we thought of reducing the peptide into smaller peptide because from industrial point of view, shorter peptide with lower production costs but similar biological activity is preferable. So we synthesized 4 smaller peptides derived from CIBR parent peptide, and we discovered that CIBR7 has similar activity to CIBR parent peptide. NMR study shows that CIBR7 peptide adopts beta turn conformation at PRGG sequence similar to its CIBR parent peptide. We are quite happy to see this result because we are able to synthesize shorter and cheaper peptide with similar structural and biological features as its parent peptide. And then we did docking study. So our docking study shows that CIBR7 peptides binds to L site, similar to its CIBR parent peptide. So our result shows that this peptide stops the movement of alpha helix 7 in LFA1 molecule, which is important for the activity of this receptor. So in short, this peptide really binds to LFA1 and stops the activity of LFA1 receptor. And next, we use HL60 cells to study how CIBR7 peptide enters the cells. Our result shows that this peptide enters the cell through energy-dependent mechanism, presumably through LFA1 receptor. CIBR7 peptide does not enter the cells indiscriminately, which proves our hypothesis that this peptide binds to cell receptor in a specific way. Right. The second part of my talk is about my research that was done in UPM after finishing my postdoc in KU. I choose to stick to autoimmune disease, but I choose different target. I trained 2 PhD students and 1 MS student throughout this project. This project is a collaborative work between UPM, EMBL-EBI, University of Edinburgh, and Surya University. My work was focused on rheumatoid arthritis. It is a chronic inflammatory disease that normally attacks joints with unknown etiology. It happens when your immune system attacks your own cells, mostly in joints. At the moment, there is no cure for rheumatoid arthritis. Current available treatment is by destroying the T cells using MTX, which brings unwanted side effects to the patient. Ibuprofen and other anti-inflammatory drugs are being used to treat the symptoms, but the root cause of this disease is not solved. Therefore, we thought we need to synthesize a peptide-based drug for treating this disease. We choose an enzyme called PAD4, or peptidyl arginine deaminase type 4, as our target. This enzyme is naturally in our body and involved in defending our body against microbial invader. This protein works by changing charged arginine residue into neutral citrulline residue. However, overexpressed PAD4 causes our own proteins to be citrullinated. Since citrullinated proteins will lose their structure due to changes in their charges, our immune system thinks that citrullinated proteins are the enemy. So here it comes. Our immune system will attack the citrullinated proteins, which will result in autoimmune disease. In this work, we designed 8 peptides based on the sequence of nucleophosphine, which is one of the natural substrates of PAD4. We discovered that PAD4 prefers peptide number 5, as you can see in this slide. And we took this peptide for the modification. As we did with our CRBR7 peptide, if you still remember the first part of my talk, peptide 5 was shortened from 9 to 7, 3, and 5 residues. Our result shows that shortened peptides are more favorable in citrullination process. And this may be due to the ability of short peptides to enter deeply into the binding pocket of PAD4. And we show that PEP57, we name it as PEP57, has the highest affinity to PAD4, so it was selected for inhibitor design. Next, we changed the arginine residue on PEP57 into furan ring based on our previous published result that small molecules with furan ring act as good inhibitor for PAD4. So we inserted furan ring in our peptide as the warhead. We call it as the warhead, which will enter deeply into PAD4 active site and inhibit the activity of the enzyme. Our IC50 study shows that DAP is the best furan containing group that can be inserted into our PEP57 peptide. The IC50 values we obtain for INH-DAP peptide is better than the IC50 value we obtain from small molecules discovered by virtual high throughput screening. Then we wanted to know how this peptide binds to its target. We did some biochemical study using very simple enzyme assay, and our study shows that our peptide binds to PAD4 reversibly and competitive with inhibition constant in micromolar range. In order to know the three-dimensional structure of our peptide inhibitor, we did one-dimensional and two-dimensional TOXI and two-dimensional NOSI experiment using proton NMR. The secondary structure of the peptide can be identified using CSI, or chemical shift index, and then intensity of NOSI cross-peak, J coupling constant of each residue, and then the structure was calculated using molecular dynamic simulation. Our result shows that our peptide, our DAP peptide, adopts a random coil structure, which is consistent with our hypothesis that this linear peptide will have random coil structure due to its great flexibility. The NMR structure was then used in molecular docking analysis. Interestingly, furan ring, the warhead of the inhibitor, enters deeply into the binding pocket of PAD4 and made contact with cysteine 645, which is the essential amino acid used by PAD4 for its activity. In addition, it is exciting to note that aspartic acid 5 in the peptide sequence form a hydrogen bond with arginine 374, which is the important amino acid in substrate recognition by PAD4. We believe that the interaction between our peptide with cysteine 645 and arginine 374 gives major contribution to inhibitory activity of this peptide. This is the last part of my presentation. I work on a project between UPM and the University of Edinburgh in the study of novel peptide inhibitor for hepatitis B virus. This work has been recently published in Organic and Biomolecular Chemistry. The focus of this work is on hepatitis B virus. This virus is a major cause in acute and persistent liver diseases in humans. If left untreated, hepatitis B may cause cirrhosis and liver cancer. We designed our peptide inhibitor based on the anatomy of hepatitis B virus. This virus has three surface antigens, namely small or S, medium or M, and large or L surface antigens. The L surface antigen plays an important role in viral entry into the host cells. The interactions between viral capsid and surface proteins are very critical for the assembly of this virus. In this work, we design and synthesize two peptides from viral surface antigens that can be used to inhibit viral assembly by blocking the association of viral capsid and surface proteins. This is the methodology we use to find the structure of our peptide using proton NMR. So basically, this is a standard protocol used in any other lab. This is the protocol we use to study the interaction between our peptides and viral capsid molecule. We use a special, beautiful NMR technique called saturation transfer difference, or STD, which can detect the differences in spectrum between unbound and bound peptide. We successfully assigned and identified all amino acids in our peptide using one-dimensional and two-dimensional Toxi NMR. From this result, we can see that the peptides are intact, stable, and not degraded. This is the result for the second peptide. And this is the final structure of two peptides. As we expected, the two peptides adopt random structures due to their flexibility. This structure was then used to study how these two peptides bind to viral capsid protein using computational docking simulation. Our docking study beautifully shows that the two peptides bind to the tip of capsid molecule in the immunodominant region of hepatitis B capsid. This is the result of STD experiment, which shows the difference in the spectrum between unbound peptide at the top in red color and bound peptide at the bottom in blue color. You can see the difference between unbound and bound spectrum. We label the peptide peaks that interact with viral capsid protein to show which amino acids are important for the binding. And this is the STD result of the second peptide, which also show the important amino acid in the peptide that interact with viral capsid protein. So, this is the last part of my presentation. I know there are too many information in my video and most of you cannot cope with it. No problem. You just have to remember four most important points in my talk. First, peptides are versatile. You can synthesize as many as peptides as you want. The limit is your imagination. Second, therapeutic peptides are increasingly popular. Please relook at the figures that I showed you before to convince yourself that therapeutic drug is very promising. Third, NMR and molecular modeling are powerful tools for peptide characterization. And last, if you need more information about my research, feel free to contact me by email, and I would be happy to have further discussion with you. Thank you.